Welcome to the Protein Synthesis series. This video focuses on translation. At the end of this lecture, students should be able to describe translation, its steps, and how proteins are developed. The genetic code is a way to represent in which mRNA codons are associated with each amino acid. There are only 20 amino acids that comprise all proteins. The diversity of proteins comes from the massive number of possible combinations of amino acids in each protein chain. The variability in length, amino acid order, and interaction between peptides of varying length and amino acid order create nearly infinite combinations. In a peptide chain of 100 amino acids, the potential combinations would be 20 raised to the 100th power. In reality, some amino acids occur more often in nature than others do, so there are fewer combinations than the theoretical possibility. mRNA is read in codons, which are 3-digit base codes. Each codon represents one amino acid. There is one codon called the start codon, which is where the DNA polymerase II begins translation of the messenger RNA. The start codon is AUG, and it codes for the amino acid methionine. Although it is not necessary to memorize the codon table, you do want to remember that the start codon, AUG, represents methionine. It follows then that each protein starts with a methionine. Conversely, all transcripts end with a stop codon. There are 3 stop codons in the codon table: UAG, UAA, and UGA. Each stop codon does not represent a separate amino acid. Instead, it just indicates stop. Translation is ended and the complex dissociates. Each mRNA codon associates through an anticodon that is part of a transfer RNA or tRNA. The tRNA is attached to an amino acid on one end of a 3-digit anticodon at the other end. tRNA runs 3' to 5', so it attaches to the transcript and maintains the antiparallel nature of the nucleic acid. The anticodon is complementary to the mRNA codon. The tRNA becomes charged when the amino acid is attached. This attachment occurs by an enzyme called aminoacyl tRNA synthetase. It is associated with that particular amino acid being coded for it. As the ribosome attaches the tRNA's amino acid to the growing peptide chain, it is removed from the tRNA. The molecules return to the cytoplasm to find another amino acid that it also codes for. Each tRNA 3-digit anticodon only codes for one amino acid. In order to determine which amino acid the codon represents, we use a codon table. Codon tables can be presented as a table format or as a pie chart. With 4 bases and 3 positions, we could have a potential of 64 combinations. With only 20 amino acids, the triplicate code is redundant, also called degenerate. The degenerate redundant code offers more than one codon that's presented for the same amino acid. For example, 3 different combinations, UAG, UAA, and UGA, all code for the stop codon. For the codon table, the column on the left represents the first base in the mRNA codon. The row across the top represents the second base in the mRNA codon. The column down the right, which is also listed at the beginning of each amino acid column as the triplet code, represents the third base in the mRNA codon. So let's practice finding GAC. The G is in the first column at the bottom in green. Moving across the top in blue, we see the A. And then the square that G and A intersect at has 2 amino acids. Finally, moving to the third base, which is the pink C, we can see our amino acid is ASP, or aspartic acid. When looking for AUG, the blue A is in the first column, the yellow U is across the top column, and the green G is in the last column, showing us that methionine, or MET, is the start codon. When looking at the pie chart, we start in the center of the rings. The innermost circle represents the first base. The next circle indicates the second base, and the outermost circle represents the third base. So let's find CUA. At the center is the yellow C. In the next ring, we see the green U. And then we find the pink A and see that this gives us leucine. Although C as the first base and U as the second base, really any of the four bases as a third base would all code for leucine. This demonstrates the redundancy found in the genetic code. Ribosomal RNA, or rRNA, is transcribed in the nucleolus, which forms around the portion of DNA that codes for that rRNA. Ribosomal RNA works with proteins to form the small and large ribosomal subunits, in addition to assembling the ribosomal complex in the cytoplasm, which creates the A, P, and E binding sites. rRNA also interacts with mRNA transcripts and recruits tRNA to the ribosome. The ribosome A site, or aminoacyl site, is where the charged tRNA first enters the ribosome. The P site, or peptidyl site, is between the A and E sites. It accepts the tRNA charged with the amino acid to add it through covalent peptide bonding to the growing peptide chain. The E site, or exit site, is adjacent to the P site, and this is where the uncharged tRNA molecule is released from the ribosome to venture out and find another amino acid that it codes for. One way to remember the site's lettering is that it spells ape when read backwards, or in the direction the charged tRNA enters the ribosome. To remember the function of sites, think A, amino acid comes in, P, peptide forms, E, empty tRNA exits. As with transcription, translation occurs in three stages: initiation, elongation, and termination. During initiation, the small ribosomal subunit binds to an mRNA strand just before the start codon at the 5' cap on the transcript, while an initiator tRNA that codes for methionine attaches to the AUG start codon. GTP provides the energy to attach the large ribosomal subunit around the transcript with the methionine-bound tRNA, completing the translation initiation complex. mRNA is read by a ribosome from the 5' to the 3' end, with each 3-base codon referred to as the reading frame or triplet. Each codon in the reading frame associates with an anticodon on the tRNA that codes for a particular amino acid. When we begin translating the mRNA transcript, it is imperative to disregard the reading frame or triplets and only focus on the individual bases until the first AUG sequence is found. From the AUG start codon, or N-terminus, because it has the free amine end, which codes for methionine, we begin translating using the triplet reading frame. As with transcription, the second phase of translation is elongation. The ribosome continues to read the mRNA codons, and the charged tRNA continue entering the A site, transferring the peptide to the amino acid in the P site, and exiting uncharged from the E site until the stop codon is reached. At the stop codon, as it is read, the entire ribosome mRNA peptide complex dissociates. The last codon before stop becomes the C-terminus, or the carboxyl end of the peptide chain. Let's practice translating an mRNA sequence. First, look for the AUG. Disregard the first 5 bases. Only begin with AUG, methionine. Now you create the triplets after AUG, your reading frames. Remember to stop translation when you reach a stop codon. So we'd have AUG, methionine, GUG, valine, AAG, lysine, CAA, glutamine, UUA, leucine, CGA, arginine, CCU, proline, GAG, glutamic acid, CUG, leucine, and UAA is a stop codon. So let's try it again, this time from the top. We'll do transcription, then translation. So we begin to use the template strand, not the coding strand, the template strand. We'll transcribe our complementary mRNA so we can synthesize. What we want to do is simply write out the mRNA chain complements first. So we would have C, A, U, G, C, G, C, A, U, A, U, G, G, C, U, G, U, A, G. Now we look for that first AUG. We don't use the triplet reading frames until we find the first AUG. And then we'll write out all the codons in the triplet reading frame and stop when we come to the first stop codon. So the first AUG means we just drop that first C base, and then we have AUG, CGC, AUA, UGG, CUG, UAA, which means stop. So we drop that last AG. So now we match the amino acids to our triplet codes. AUG is methionine. CGC is arginine. AUA is isoleucine. UGG is tryptophan. CUG is leucine. And then we have the stop codon. So that ends the amino acid. Students should be able to understand the central dogma of biology. Students should be able to describe the 3 stages of translation. Students should be able to explain the structure and function of ribosomes. Students should be able to transcribe DNA into mRNA and translate it into a peptide chain. Students should be able to differentiate between RNA, mRNA, tRNA, and rRNA. Students should be able to use the codon table and the pie chart to identify the start and stop codons, as well as how to read the amino acid codes.